are divided into two groups. Okay. What are those two groups of the fungi? As for Ainsworth classification, fungi are divided into two groups. What are those? I already told you. Myxomycota and Eumycota. Myxo means false. False fungi, and U means true or yes. True fungi. Okay. Fungal kingdom are divided into two, that are false fungi and true fungi. Myxomycota and Eumycota. Myxomycota and Eumycota. Okay. Myxo, false. That means it are looking like a fungi, but it having the plasmodial-like structure, amoeboid-like structure. Plasmodium. There is no specific structure. There is no cell wall component. There is no specific structure. It looks like an amoeboid structure. It having the plasmodium, gelatinous body. It having the gelatinous body. Okay. And that all are calling it as Myxomycota. The plant body of Myxomycota is calling it as plasmodium. That is also very important. Plant body of the Myxomycota is calling it as
Plasmodium.
What is the basic structure unit of the fungi? What is the basic structure unit of the fungi? Hyphae. You have studied. Basic structural unit of the fungi is hyphae. Okay, hyphae, mycelium, we have studied. Mycelium, what does it mean, the mycelium? Hyphal network, interconnecting of the hyphae. Interconnecting of the hyphae are forming as a networking structure, net-like structure is called as mycelium. Okay. And then Eumycota, which having the true fungal character, which having the true cell wall, which having the true fungal, all the remaining fungal characters. Okay. And this Eumycota fungi thallus structure, we calling it as hyphae. Whereas the Myxomycota thallus structure, we calling it as, so many times asked the question, basic structure of the Myxomycota, plasmodium. What is it?
Plasmodium.
Yes. Okay. And then Myxomycota again divided into how many?
4.
4 divisions. How many divisions?
4 divisions.
No, 4 classes. Myxomycota division is divided into 4 classes. What are those 4 classes? Read out. Acrasiomycetes, Hydromyxomycetes, Myxomycetes, and Plasmodiophoromycetes. Okay. These are the 4 types that we will discuss elaborately. Not elaborate, we only briefly. Okay. And then we will next Eumycota are divided into Eumycota, true fungi.
5 classes.
What are those? Not 5 classes, here subdivisions. Again, it are divided into classes, orders. Okay. Here it are divided into Eumycota, it divided into how many?
5 subdivisions.
5? Yes, 5 subdivisions. What are those?
Mastigomycotina, Zygomycotina, Asst.Mycotina, Mycotina, Deuteromycotina.
Yes. Okay. You read out so that you can, your brain will be activated. Otherwise, if you passive, it may not reach to your brain. Okay. Mastigomycotina, Zygomycotina, Ascomycotina, Basidiomycotina, and Deuteromycotina. Okay. Based upon their sexual or asexual spores, based upon their reproductive characters, based upon their reproductive character, divided into 5 groups. Okay. Mastigomycotina, Mastigonymphs, that means flagellas, flagellated hyphae are zoosporic fungi which are in the flagella. See, flagella 1, 2. Flagella, this is the fungal spores. Fungal spores, if it having the flagella, that are all the fungi, if it having the flagella, if the fungal group which having the motility, movement, chalanam, movement through the flagella. That all type of the fungi are grouped in Mastigomycotina. In which group? Mastigomycotina. Mastigomycotina again divided into 3 classes. What are those? Already explained you. Chytridiomystis, Hypocytridiomystis, Oomystis. That we will discuss. Huh? And Zygomycotina. The sexual spores are zygospores. The sexual spores are zygospores. If the sexual spores are zygospores, that we call it as Zygomycotina. That means you know the mucorrhizopus you have studied, na?
Yes, sir.
In the mucorrhizopus you have studied 2 opposite strains. Gametangias comes together and it attached and it form the fruiting body like structure, round globular structure with appendages by melting the 2 cell walls of the 2 opposite plus and minus. You know the what we call in that? This also question raising from this also. Heterothallism. Have you heard the word heterothallism?
Yes, sir.
What does it mean, the heterothallism? Who discovered first?
2 different thallus.
2 different thallus. If the thallus having the various different type of the genetic material, different strain, plus strain and minus strain, that we calling it as heterothallic. Heterothallic. You know, dioecious, monoecious, unisexual, bisexual.
Yes, sir.
What does it mean the bisexual? What does it mean? What does it mean?
2 opposite gametes fusion. Bisexual.
Bisexual, huh? Bisexual, unisexual. If the 2 opposite strains, 2 opposite sex are present in the same thallus, that is bisexual. If it's a male and female, Anthridia and Archegonia are present in different thallus. That we call it as unisexual. That is nothing but heterothallic. That is nothing but dioecious. You know dioecious, monoecious. Mono means
One.
monoaction. Huh? Mono means monoaction. Huh? Silence, huh? Or monarcha. Mono means single. Yes. If that means the single thallus with the two opposite strains, male and female gametangia are present. That we calling it as monoecious. If the male and female plus strain separate thallus and minus strain separate thallus. If it is there, that we call dioecious. That is nothing but heterothallism. That is nothing but heterothallism, plus strain and minus strain, two different strains. Two opposite strain mycelium, if it is coming together and mating, that we are calling it as heterothallism. That is first discovered by A. Blacksley. Who is that? This question raised so many times in so many competitive exams. A. Blacksley. A. Mucormycido fungi. Zygomystis fungi. Heterothallism. Okay. That zygospores are formed in the zygomycotina. Which fungi it is forming? The zygospore, that we are calling it as zygomycotinous fungi. And then which group of fungi which having the ascus, that means ascus fruiting body, and which release the ascospores, that all comes under ascomycotina. Which group of fungi which having the button-shaped fruiting body, tertiary mycelia, or button-shaped fruiting body, basidiums, that having the spores, basidiospores, that are grouped as basidiomycotina. That are grouped as basidiomycotina. And you know the deuteromycotina? In which group of fungi sexual reproduction is absent or not known yet? Sexual reproduction of fungi is not there or not known yet. In this fungi, that are grouped as deuteromycotina. You know the deuteromycotina fungi is also called as imperfect fungi. What it is? Imperfect, not the perfect, like you. I think you are not perfect in your subject. In which subject you are perfect? In which aspect you are perfect? Just going college and doing enjoy. Or staying at home is imperfect. You are perfect in staying at home. In which aspect you are perfect? Sultana? Ruchita? Huh? Okay. Leave it. Huh? Deuteromyces fungi, also called as imperfect fungi. What does it mean? What it is?
Imperfect.
Imperfect fungi. We also calling it as imperfect fungi. Deuteromyces fungi, also called as imperfect fungi, where there is no perfect stage. You know the perfect stage means? Perfect stage. Sexual reproduction. If the sexual reproduction stage is there, that is perfect stage. If the sexual reproduction is absent, that we call it as imperfect. Imperfect, not perfect. That means perfect stage is absent. Sexual reproduction is absent or not known yet. That all the group of fungi are included in the Deuteromycotina. Perfect stage is absent. That's why we call Deuteromyces fungi as imperfect fungi. But it is imperfect, not perfect. Imperfect fungi. Okay? That's the question. Which among is called as imperfect fungi? Mycomycotina, Zygomycotina, Asst.Mycotina, Bacillomycotina, Deuteromycotina.
Deuteromycotina.
Yeah, Deuteromycotina is called as imperfect fungi. Why?
Sexual reproduction is absent.
Yes, sexual reproduction, perfect stage is absent. And you know which group of fungi is called as dustbin fungi? There are so many times. Which group of fungi is called as dustbin fungi? You know the dustbin?
Yes, sir.
Dustbin fungi. Deuteromycotina fungi is calling as dustbin fungi. Deuteromyces fungi is also called as dustbin fungi. Deuteromyces fungi is called as imperfect fungi and dustbin fungi. Why are we calling it as dustbin fungi?
No
The
sense.
scientists, mycologists, they consider it as dustbin fungi. It is not the perfect group of fungi. Whenever they identified first any fungi, they kept in this group, in this subdivision. But it is Deuteromycotina. If the sexual reproduction stage identified, then based upon the sexual reproduction stage, whether it having the motile spore, or whether it having the zygospore, or whether it having the ascospore, or whether it having the basidiospore. If it having the motile spores, they shift from Deuteromycotina to Asst.Mycotina. If they having the zygospore, zygosporangium, they shift to Zygomycotina. If they are having the ascus bodies, fruiting bodies, and ascospores in the sexual reproduction, the shift from Deuteromycotina to Asst.Mycotina. If they are having the button-shaped fruiting bodies, they shift from Deuteromycotina to Bacillomycotina. If not found any of these four type of the spores, again it will be remain in the Deuteromycotina. You know, Aspergillus, Penicillium, like there are so many species first kept in Deuteromycotina. After identified the perfect stage, telomorph. You know the perfect stage is called as, competitive exam so many times asked the question. Perfect stage of fungi is called as telomorph. Perfect stage of fungi is called as
Telomorph.
telomorph. Imperfect stage or asexual stage of fungi is called as, I already told you.
Deuteromycotina.
No, I already told you 3 states of fungi. Anamorph, telomorph, holomorph. These 3 states of the fungi I explained you earlier. In the Thales organization we discussed asexual stage of the fungi is called as anamorph. You should remember from this also, every time question is raising. Asexual stage of fungi, anamorph. The perfect state of fungi is called as telomorph, holomorph, both. Anamorph, telomorph is commonly called as holomorph. Okay. And you know what is the perfect stage of Aspergillus? So many times asked the question. What is the sexual stage? Perfect stage of Aspergillus. What is the telomorph of Aspergillus? What is the telomorph of Penicillium? So many times they ask the question. There are so many exams. Don't you know? I already explained. In the reproduction chapter, I already explained. You should remember the perfect stage of Aspergillus nidulans is called as Emersella nidulans. What it is? What it is?
Emersella.
Emersella. Okay? Emersella nidulans. Perfect stage of Aspergillus is Emersella nidulans. You should remember. And perfect stage of penicillium. You know the penicillium? Penicillin? TT injection, penicillin. Alexander Fleming. You know the Alexander Fleming?
Yes, sir.
How you know? Have you seen? Have you seen Alexander Fleming? Or have you met Alexander Fleming? How do you know Alexander Fleming?
Photos.
As he discovered the penicillin. You know, as he discovered the penicillin antibiotic from the Penicillium notatum. Yes or not? You know that only. Yes? When Alexander Fleming discovered the penicillin from the Penicillium, this question also raised so many times. Penicillin is prepared from which species? Which fungi? Muca, Rhizopus, Aspergillus, Penicillium. Like that, they are the question. Or from Penicillium chrysogenum or Penicillium notatum, Penicillium inundatum, Penicillium, like that, so many species, especially from Penicillium notatum. Okay, that also you should remember. The perfect stage of Penicillium is Toleromyces. Perfect stage, telomorph of Penicillium is Toleromyces. After identifying the sexual stage in the Penicillium, they given the name as Toleromyces. What name it is? Telomorph name. Name of the telomorph is Toleromyces. Name of the telomorph of Aspergillus is Emersella. Emersella nidulans. Okay? Like that, if the perfect stage is identified, then it shifts to any other 4 groups. If the perfect stage is not identified or not known, then it will be remain in the which group? Deuteromycodina group. Deuteromycodina group, dustbin fungi group or imperfect fungi group. Okay. Dustbin fungi group or imperfect fungi group. Okay. This is all the basic knowledge you should know. Okay. Are you listening or not?
Yes, sir.
You are getting bored. You are listening my class 1 hour. Huh? So you are getting bored.
No, sir.
Yes? Yes. You are getting bored.
No, sir.
You have to take only half an hour or 40 minutes. We can't stay this much of the time. Yes?
No, sir.
Okay. You know, false fungi and true fungi. Myxomycota and Eumycota. Myxoma is false. That Myxomycota we also called it as false fungi, slime molds. False fungi, slime molds, Which group of fungi we called as false fungi or slime molds? Which group?
Myxomycota.
Yes. Okay. Myxomycota. You see, what are the important features they have taken consideration in the classification? I already told you at the first. What it is, either it is autotrophic or heterotrophic, either it is free-living or parasitic, based upon the nutrition. Based upon the nutrition. Okay, and cell wall composition, reserve food material, and cell structure, either it is unicellular or filamentous. These four characters. What are those four characters? Cell wall composition, nutrition, reserve food material, and cell structure. You should remember what are those four characters. Take in consideration the classification of the fungi. Asked so many times. They may give any total four or give any two, and they give the choice A and B or B and C or A and C, A and D. Like that, they are the question. If you know the four, then only you can write the correct answer. You can take the correct answer. What are those four basic characters? Cell wall composition, reserve food material, cell structure, and nutrition. Okay, have you understood?
Yes,
and
sir.
then, okay, then you see what we have told you, the false fungi and true fungi. False fungi, Myxomycota or slime molds, are divided into four classes. How many classes?
4 classes.
What are those? What are those? You read out. Class 1,
Acrasiomycetes.
Class 2,
Hydromyxomycetes. Class 3, Myxomycetes. Class 4, Plasmodiopharmycetes.
Yeah. What are the difference between? You see, if it is a slime mold, gelatinous structure, if it is a cellular, if it look like a cells attachment like structure, if it is a cellular gelatinous structure, that all groups in the acrasiomycetes. Or if it is forming a network like structure, slime mold, the plasmodium, the slime mold, if it is forming a network like structure, net like structure, connecting link, network like structure, that we grouped in the hydromyxomycetes. Actually, true myxomycetes are like a plasmodium structure that are grouped in myxomycetes. And parasitic fungi, parasitic slime molds, endoparasitic, internal parasitic fungi, parasitic molds are grouped under BSc BJET, BSc Microbiology. Which group you are? What is your group in degree?
BZC.
BJET. Ruchita?
BSc BJC, sir.
BJET. Okay. You see, what are the differences between in the Myxomycota? If the cellular structure mold that are acrasiomycetes. If the network like molds that are hydromyxomycetes. And true slime molds are like a plasmodium that are myxomycetes. And parasitic slime molds are called as plasmodiophoramycetes. Okay. And then the next is Mycota, true fungi. I already told you. I already told you what I told you. Divided into 5 groups. What are those 5 subdivisions? Masticomycotina, Zygomycotina, Asst.Mycotina, Basidiomycotina, and Deuteromycotina. Okay. The first one is Masticomycotina. What I told you, fungi which having the flagella. Fungi which having the flagella. You see the flagella. Fungi which shows the motility, movement. Fungi which having the zoospores, that means motile spores. Motile spores are grouped in Masticomycotina. Okay, and then if it having the only one flagella, that is smooth, one flagella, that is smooth flagella, that type of the zoospore is fungi, unicellular zoospore fungi are grouped in Chytridiomystis. If the one flagella, that is tensile type, having the pimbrils, feathery, airy-like structure. If the flagella having the air-like structure, pimbril-like structure, that grouped as Hypochytridiomystis. Okay, and if the zoospores having the two flagella, not the single flagella, two flagella. One is whiplash, one is tensile. If the two type of the flagellas are there in the zoospore, that grouped in which class? BA class. And then BCom class. Are you in BA class or BCom class? Huh?
No, sir.
So you should respond. If the two flagellas are present, that group in the Eumystis class. Which class? Eumystis. Only one flagella, that is tensile type, that are grouped as Chytridiomystis. One flagella tensile type, that grouped as Hypochytridiomystis. One flagella whiplash type, that means Acromystis type. You know, acronematic and pluronymatic. Okay, and the next is Zygomycotina, which having the zygospores. You know the Zygomystis fungi, thallus, hyphae, we calling it as Cenocytic thallus. You know, I already told you, thallus organization. Cenocytic, that means there is no septa, that means more nucleus are present in the cytoplasm. More numerous. Cenocytic. More nucleus, numerous nucleus are present in the thallus. Why? Because there is no septa. There is no cross wall in the hyphae. Aseptate hyphae. And which having the zygospores are grouped in Zygomycotina. Okay, Zygomycotina again divided into two classes, that are Zygomystis, Trichomystis. Zygomystis and Trichomystis. What type of the fungi grouped in Zygomystis? Mycelium, if it is inside the host, if suppose it is the host tissue, then inside the host tissue, if the mycelium is present, that grouped in Zygomystis. Okay, if the mycelium is upon the host, outer side of the host, or peripheral of the host, or nearby the host, that grouped in trichomycetes. That grouped in trichomycetes. Okay, have you understood?
Yes, sir.
And then the next is ascomycetes, which having the ascospores, septate hyphae, that especially simple porous septate hyphae, which having the simple porous, there is a cavity hole, porous space in the septa. Simple porous septa is present in ascomycetes fungi. Aseptate fungi is present in zygomycetes fungi. Okay, ascocarp. What are the types of the ascocarp? Ascomycotina fruiting bodies type. I already told you. Clistothecium, perithecium, apothecium, ascostroma, pseudothecium. These are the 4 types of the ascocarp. You see, Ascomycotina again divided into
6 classes.
6. 6 classes. What are those 6 classes?
Hemiascomycetes, Locuoscomycetes, Plectomycetes, Lobule, Beniomycetes,
Labalbino.
Discomycetes.
Okay. Perinomycetes. Who read out the perinomycetes? Your grandpa. Your grandma will read out.
Sorry, sir.
Okay. These are the 6 classes that are hemiascomycetes. That means which having no ascocarp, no ascus fruiting body. Ascus fruiting body is option. Ascae, direct ascae are naked, not in the fruiting body. That are grouped in hemiascomycetes. If the fruit body is as ascostroma, that means pseudothecium. That grouped in laculascomycetes. Okay, and if the fruiting body is a clistothecium, round, globular, thick-coated, without porous fruiting body, that group of fungi is grouped under Plectomycetes. Okay, if the fruit body is perithecium, round, globular, open with the simple porous, small porous space, fruiting body, that we call as perithecium. Ostiole, porous space is present. That porous space we calling it as ostiole, mukarandramu, ostiole. That ostiole present fruiting body we calling it as perithecium. That perithecium fruiting body fungi are grouped in Labalbinomycetes. Labalbinomycetes. Okay. And same perithecium fruiting body as like perithecium fruiting body, and these are not parasite upon the arthropods. With the parasites upon arthropod, that group as Labalbinomycetes. If the same fruiting body, if it is not on parasite and arthropods, insects, that group as Pherinomycetes. Actual true Ascomycetes, fruiting body is Apothecium. Saucer shaped fruiting body, which having the saucer shaped fruiting body are grouped under Discomycetes. You know, Pezaiza Apothecium. You know, Pezaiza, what type of the fungi it is? Where it grows? Capropilus fungi. Do you know the Capropilus fungi?
No, sir.
Capropilus means it grown on dung, cow, animal dung, cow or whatever it is, the kettle dung. Capro means excretory material, dung. Pilus means love, which grown on dung. That we call it as Capropilus fungi. Okay, the Capropilus fungi which having the apothecium, saucer-shaped fruiting body. Pezaiza is a Capropilus fungi. This question also raised so many times. Which among is the Capropilus fungi? Okay, which among is the insectivilus? That means grown on insects. Entomophilus. Entomophilus fungi. Grown on entomos, insects. Okay, the next is Basidiomycotina. You know, yeast or septate mycelium. What type of the septa is present in the Basidiomycotina? I told you. Dalifor septa. కొబ్బరి చిప్పలాగా తెచ్చుకున్నట్టుగా రెండు ఉబ్బినటువంటి బాడీలు ఉంటాయని చెప్పా. In the cell, paranthrosomes, 2 paranthrosomes. This type of the porous septa is present in Basidiomycotina. That we calling it as Dalifor septa. This question also raised so many times. Dalifor septa is present in Asst.Mycotina, Basidiomycotina, Dr.Mycotina, Asst.Mycotina, Zygomycotina. Which group? Basidiomycotina. Okay. Basidiospore, which having the basidium and basidiospores, that all grouped in Basidiomycotina, button-shaped fruiting bodies. Again, this is divided into
3 classes.
what are those?
Teleomycetes, Hymenomycetes, Gastromycetes.
Gastromyces, not gastro, gastro.
Gastro.
G, not C. Okay, gastromyces. Okay, these are the 3 types. You see, teleomyces. Here, the basidiocarp is option. It having the teleospores. You know, basidiocarp 2 types I told you. Pragmobasidium and holobasidium. Fragmented. If the basidium is fragmented, basidios is fragmented, that we call it as just now I told you now, holobasidium and holobasidium and pragmobasidium. If it having the septa fragmented. In the fragmented septa, basidiospore, it form the teleospores, also called as promycelium, probasidium. Okay, and then the next is Hymenomycetes. You see, the basidiocarp is present. In the basidiocarp section, it having the 3 parts usually in the fruiting body: excipulum, subhymenium, and hymenium. This question also raised so many times. Which among is the fertile part in the basidiocarp? Excipulum, hymenium, or subhymenium? Hymenium is the fertile part. In the hymenium part, the spores will be generated. Even in the spores or basidiospores that generate in the hymenium, from the hymenium layer. Hymenium layer is a fertile layer. From that only the spores, sexual spores will come out. That's why the fertile part of the fruiting body is called as called as hymenium. It's called as hymenium. Exipulum is the vegetative part. Cybohymenium is the in between the exipulum and hymenium, in between the fruiting body and vegetative thallus. Okay, and the gastromystis. You see, basidiocarp is present. Hymenium is enclosed inside the basidiocarp, not at outside like this. Hymenium, that means fruiting body structure, the spore structure, where the spores are inside, endosporous. This is not releasing by water dropping mechanism. You know the Buller's phenomena. Buller's phenomena. That question also raised so many times. Buller, not Buller, Buller. Water drop mechanism of spore dispersion in the basidiomycetes is called as Buller mechanism. Spores, basidospores released by touching the water drops upon the basidiocarp. That is Buller mechanism, asked so many times this question. Okay, and the last is Deuteromycotina, imperfect fungi or dustbin fungi. Perfect stage is absent in this fungi. Again, these are divided into
3 classes.
what are those?
Blastomycetes, hypomycetes, coelomycetes.
Yes, blastomycetes, hypomycetes, and coelomycetes. Okay. You see, pseudomycelium is present. It looks like a white networking like structure. Whether it is which group, which type of the fungi mycelium, we can't distinguish. That we call it as pseudomycelium. Not as like a networking mycelium. Pseudomycelium like structure. This is not well developed. Either it is a single cellular like structure or false networking structure. That group are blastomycetes. And if the mycelia having the sterile, if the mycelia having the only asexual spore, either arthrospores, conidiospores, chlamydospores, conidiospores, arthrospores, blastospores, iodias, arthrospores. These are the types of the asexual spores in the fungi that you have studied in the reproduction. These spores, this having the sterile mycelium, and it form the spore structure, spores formation, asexual spores. Okay, either conidiospore, ballistospores, arthrospores, iodia, Accidia, iodia, chlamydospores, whatever it is a spore type, either endogenous or exogenous spore type, that are all grouped in hypomycetes. Most of the Deuteromycetes fungi comes under hypomycetes. If the asexual spores are formed in either pycnidium, simple porous round fruiting body, or in acervulus, saucer-shaped fruiting body. Asexual spores, that means pycniospores or esiospores. Pycniospores or esiospores, like that. Spores, if it having, that are grouped under coelomycetes. Okay, these are the 3 types of Deuteromycotina imperfect fungi. Okay, based upon pathology also divided You know, I already told you the lower fungi also calling it as phycomycetes. Why? Because these are look like algae. Phyco means algae, phycology, phycology, algae, study about the algae. It look like an algae. Rhizopas, mucor, albugo, pythium, Phytoptera. These all are look like algal structure. That's why we calling it as phycomycetes, which having the sac, successor recruiting body grouped and ascomycetes. If it having the button shape club, gada, club shape protein bodies that are grouped in Bastomyces. If they don't having any perfect state, that grouped and Deuteromycotina. Example, Cercospora personata, Cercospora arachidicula, you have studied. Ticca disease of groundnut. Coltotrichum, you know the Coltotrichum palcatum, it causes red rot of sugarcane. Trichoderma, we are using as a biopesticide. Pericularia, Fusarium, Trichoderma, these we are using as a biopesticide. Agaricus, polyporus, this we are using as a mushrooms. Puccinia, you know, Puccinia you have studied. Wheat rust disease. Ustilago, Cystopus, you have studied white rust. Lycopodon is the mushroom, macrofungi that having the medical properties. You know, in Ascomycetes yeast you have studied, Aspergillus you have studied, now Penicillium you have studied, Neurospora that we are using as a genetic study material of the fungi. Competitive exams, so many competitive exams they ask. Which fungi using in the genetic study or molecular study, which group of fungi, which fungi? That is Neurospora crassa. Neurospora, this one. We are using as a genetic experimental organism, as like Drosophila in the animals. Drosophila we are using as a genetic experimental organism. Okay, and then you know this you should know, though it is not there in your syllabus. In the PG, you will study. This is the phylogenetic classification of the fungi. As a phylogenetic classification, you know what does it mean? The phylogenetic classification based upon the genetic study, sequencing, DNA and RNA sequencing. Classification based upon so many branches, not only the morphological characters. Along with the morphological character, cell biology, cytology, physiology, anatomy, embryology, the remaining science data also combined along with the morphological study, that we calling it as phylogenetic classification. You know, Engler and Prandtl classification, Hutchinson, Thaktajan, Conquest classification in the angiosperms are called as phylogenetic classification. That question also raising so many times. Which among is the phylogenetic classification? Engler and Prandtl, Hutchinson, Thaktajan, Conquest, Bessate. These all classification are called as phylogenetic classification. Here, the Uptal classification is called as phylogenetic classification. Uptal means association of fungal taxonomies. Association of fungal taxonomies group. Head in this group is Hibbett. That's why you also called as Hibbett classification. There, they categorize based upon phylogenetic tree. They divided the fungi into various groups. You see, Dichoria is the higher fungi, Ascomycota and Basidiomycota. And the remaining is Glomeromycota, Chytridiomycota, Mycology, and Blastocladiomycota, and Microsporidia. Divided into how many? 1, 2, 3, 4, 5, 6 groups. Okay. As for the phylogenetic classification, Hibbett classification, Uptal classification. Okay, you should remember this classification. And the latest classification of fungi is Tedesco classification. Who is that? Who is that? Leo Tedderson classification. There is a high-level classification of fungi, latest phylogenetic classification of the fungi. Okay, that if you select in the MSc botany, in the PG botany, you will study these two classifications. Okay, up to degree you have studied only Ainsworth classification. Okay, this is the classification of the fungi. Okay, that we have studied. The very important. So many questions raised from this. Okay, you go through again in this video. You will get more question answer from this classification of the fungi. Okay, whose classification we are following, what type of the classification it is. Okay, what characters we take in consideration for the classification of the fungi, how many groups of they divided into how many divisions of the fungi, how many subdivisions of the fungi, how many classes of the fungi, in which fungi plasmodium like plant body is present, in which fungal group flagellas are present, in which fungal group asca is present, in which fungal group basidium, stool, puffballs, mushrooms, jasters are present, which group of fungi is called as macrofungi, which group of fungi is higher fungi, in which group of fungi zygospores are present, or muca rhizopus belongs to which group of fungi, or penicillium belongs to which group of fungi. Like there are so many questions will raise from this classification of the fungi. Okay, with this I'm concluding almost 2 hours. I have so many works, other works, book writing works, these are pending, and the PG classes work also pending. So many works are being penned. Okay, so you go through these, you follow, you again watch this video, and you will get more answer in the competitive exam point of view, in real life point of view, and the enthusiastic point of view of the botany. You will learn, you will get some amount of perfection of the knowledge in fungi. You feel proud as a botanist. Okay?
Yes.
These all are the classification of the fungi. This we already discussed as for Alexopoulos and Mims classification. This we have studied already. This is a Ainsworth classification. What we have studied. Brief outline. Mycology 3 classes. Zygomycology 2 classes. Asst.Professor 6 classes. Basidiomycology 3 classes. And Deuteromycology 3 classes. This is the outline of the classification of fungi as for Ainsworth classification. Okay, this all we have studied. Okay. Okay. Right.
Okay, my dear friends, is there any doubts?
Any doubt?
Ruchita?
Any clarification? Any doubt? You again, once again, you go through this video,
No
then
doubts.
you will get an idea.
Okay.
At a time, you may not get the total knowledge. If you again listen this, you will get a lot of clarification. Your brain will be optimized. Your thoughts will be full power on fungi, so that you will remember for the long time. Okay.
Okay, sir. Thank you very much.
longest class, nearly 2 hours.
Excellent session, Nagaru. Thank you very much. Okay, shall we going to wind up?
Yeah. Okay.
Thank you.